I will share my PPT now. Yeah, this is the anther. What is the anther? How to development of anther? Mm, and what are the importance of anther? These are things we will discuss. The types of content, anther, introduction of anther, structure, development of the anther and pollen, anther wall, epidermal, endothecium, middle layer, tapetum, amboidal tapetal, secretory tissues, tapetal, orbiculus, functional of orbiculus, tapetal membrane, function of tapetal. These are things. First one, what is the anther? What is the anther? What for the importance of anther? Here, stamens are the male reproductive organs of flower plants is called the anther. All stamens is called the anthers. The anther, they consist of an anther. The site of the pollen development and dispersal. The anther is born on a stalk-like filamentous that transmit water and nutrients to the anther, and also position is it to aid pollen dispersal. The anther dehiscence at maturity, at maturity in most of the angiosperms plant. Most of the angiosperms, angiosperm plants by angiosperm by the longitudinal slit, the stomium to release the pollen grains. The pollen grain represent the highly reduced male gametophyte of flowering plants that are formed within a sporophytic tissue of the anther. These microsporogenesis of pollen grains are the carriers of male gametes or male sperms of the cell that play a central role in plant reproduction, reducing during the process of double fertilization. This diagram you can observe. This is a total flower. In the flower, this part, green color part is the gynoecium. These are andrecium. Gynoecium, to provide the gynoecium is there, that is called the carpels. Andrecium is called the stamens. Gynoecium you can observe. Ovules are megasporangium. The total name is called the ovary. After ovary, upside in the upper part you can observe stigma is there, style and ovary. Stigma, this one, this is the style, this is the ovules inside. This part is called the carpel, sorry, ovary, and the can is called the carpels. Total. Another one, this is the andrecium. Andrecium is called the stamens. Stamens here, inside the stamens, pollen grains are present. And this pollen grains name is called microsporangia. These are all anther. This is the filament. This is the stamens. This is the female gametophyte. This one is the male gametophyte. See, you can observe these are the stigma, filament. See, this is the stigma part. Stigma, this is the style. You can observe the petals. These are the anther or filament body. See, this is the filament and stigma, style. See, this is the anther. This is the stigma, filament. So this is the style part, stigma part, filament. This part is filament, anther, this one. institution. This is for the hibiscus flower stamen showing monothecus, are like this. Different parts is the different flowers, different anther as the observed in the different areas. See, the structure of anther. A typical anther is bilobed structure. A typical anther is called bilobed structure, dithecous structure, dithecous structure with two microsporangium in each lobe microsporangia, each lobe. Therefore, an anther is a tetrasporangiate structure with four microsporangia. See, this is the four-lobed structure is there. In this four-lobed structure, microsporangia and non-sporogeneal tissues that join together anther lobes. This is called the anther lobes. These anther lobes, this total is called the anther lobes is known as the connectivity tissues, connectivities. See, this 1, 2, 3, 4 lobes are there. Each and every lobe inside to connectivity is present. This is the anther wall. Outer wall is the epidermis, anther wall, and inner side is the middle layer. That is the sporogenous tissue, and tapetum layers are there. Microspores are present. And a single vascular strand. This one here, you can observe this one, this layer. This layer is called the single vascular strand is embedded in the connective in each lobe. The two microsporangia are separated by a strip of sterile tissue. The intersporangial septum in a mature anther, the two sporangia in an anther lobe become conflict due to the enzymatic tissues are present. This one is the picture of anther. See, epidermis layer, endothelium, middle layer, connectivity tissues, all are the present in the anther tissue. See, this anther, epidermis, androsporangia, all are the present in the anther tissue. And one second. The anther, main important of the anther, the male gametophyte development is the anther tissue. This one, the main source of the anther is called stamens. Sometimes single-lobed stamens are there. Sometimes doubled layered stamens are also present. This one is the this one is the present into the anther-lobed structure. And mm, see here, a typical anther has four microsporangia. Always anther name is called stamens or microsporangia or pollen grains or male gametophyte. These are the names, the same one. A typical anther has four microsporangia. That is called tetrasporangiate. Two in each lobe. One of the easiest method of studying structural details anther by causing transfer section of anther. This is the transfer section of anther. You can observe to the four layered of the structure is present. The first layer is the epidermis layer, the first layer. The second layer endothelium. The third layer, middle layer, fourth one is called the tapetum layer. This is the diagram represent a typical anther structure and you can observe to the in the transfer section of anther. See this the second picture enlarged to show basic cell layer tissue. This is the endothelium. This is the middle layer and microspore mother cells. And inside is the tapetum cells also present. And how to develop the anther? A young anther consists of a homogeneous mass of meristematic tissue. In the starting, anther, how to develop? Where the development is to start, in that part, the meristematic tissue is present. Here also, the young anther consists of the homogeneous. Two types of tissues, you know, homogeneous tissue, heterogeneous. Here consists of a homogeneous mass of meristematic cell that are surrounded by the epidermis. See, the anther transfer section, you can observe to the four-lobed structure where the epidermis is there. In that epidermis part, the homogeneous meristematic tissues are present. As the anther develop further, it begins assume a four-lobed structures are you can observe and appearance. And one are a group of two or more hypodermal archisporial cells. Two cells are there. One is the archisporia, meaning that cells are to development of the antheridia, archisporium cells differentiated in the each lobe. Thus, these cells appearance conspicuously different from rest of other cells. These other cells are very large in size, dense cytoplasm, prominent nuclei is there. Which one? Where the archisporial cells are there? In that archisporial cells, you can observe to the in the normal tissue cells or normal cells and archisporial both are present inside the anther. But how to identify the archisporial and normal cell? Where the archisporial cells are there, large in size, dense in cytoplasm, prominent nuclei is there. These all cells name is called archisporial cells. Now these archisporial cells may be either a single layer or made up of sometime very several vertical layers, rows of cells are present. Did you understood? No, archisporium cell, each and every archisporium, either single layered, some is the single layered are there. Some archisporial cells, several vertical rows of the cells. The later in a transverse section of anther appears as a plate-like cells. These archisporial cells divide in a plane of parallel to the outer wall of the anther lobe. Such divisions is called as periclinical division. Such division name is called periclinical division. The first division is called the periclinical division. After one such division, the primary pericytial cells towards the primary pericytial towards the epidermis abbreviated as per towards the abbreviated as the PPC the present. This one is the PPC as the present in this one. Archisporial in this one towards the intervallar of the This one is the present in these archisporial cells. After that one, you can observe to the formed in the PPC by repeated periclinical cell. The primary sporogenous cells or primary pericytial cells. Both after the one division, where in the anther lobe, the archisporial cells, the first archisporial cell, first division is the plain parallel division to the outer wall of the anther lobe. Where you can divisions are there? Where the outer layer of the anther lobe is there, in that part, first cell division is the parallel to plane of the parallel. Such division is called pericytial division. After one such division, one pericytial cell, where they can that one towards the epidermal, abbreviated, that is called PPC. PPC meaning pericytial cell, PSC meaning pericytial sporogenous, primary sporogenous cells towards the inner side of the anther sac. Observe abbreviated, that is called PSV. Primary pericytial cells, primary sporogenous cells. This is called the PPC, PSC. Are formed the PPC cell, primary pericytial cells, by repeated periclinical and anticlinical. Two divisions are present. Periclinical divisions are there. Anticlinical divisions are there at right angle to the surface. Gives rise to, after divisions, to form to the 2 to 5 layers of the anther wall. And the cell of the primary sporogenous layer, either directly or after a few mitotic division, directly or after a few mitotic divisions, to form the microspore mother cell. That is called MMC. MMC, microspore mother cell. The schematic representation of the antogeny of anther wall layer and the microspore mother cell is presented. Now the final MMC is present. That MMC is called the microspore mother cell. The final to the form the microspore mother cell. This is the undifferentiated anther. One is the epidermis. Second one is the archisporium. This is the archisporium can be divided into primary pericytial cells and primary sporogenous cells. The archisporium is present into the primary pericytial cells and primary sporogenous cells. These primary pericytial cells again can be divided into the secondary pericytial cells. Secondary pericytial cells again to form endothelium upper middle layer. Secondary lower middle layer, middle cells, tapetal cells. Again, these are two can be endothelium is endothelium cell, upper middle layer cells, lower middle layer, tapetum. This is the primary PPC, primary pericytial. Next one PSP, primary sporogenous cells. The secondary sporogenous cells, microspore mother cell to form. What is the anther layers? The wall mature of the anther consists of following 4 layers. Epidermis, epidermis, endothelium. The first layer name is called epidermis. The second layer endothelium. Third one is the middle layer. Fourth one is called the tapetum. The first one is epidermis layer. The epidermis is the outermost layer of the anther wall. It undergoes only anticlinical division. This epidermis is the outermost layer, and anther wall the undergoes only the anticlinical division. In this manner, it is able to crop up with the rapidly enlarging internal tissue. In a mature anther, the epidermal cells appear greatly scratched and flattened. The function of the epidermal cells is mainly to provide protective overcoming to the internal tissue. This is the first layer is completed. The second one is the endothelium. The layers of the cells lying immediately next to the epidermis is the endothelium, which is responsible for the dehiscence of the anther. This is the most important for the dehiscence of the anther. This cells is called endothelium, which is responsible for the dehiscence of anther. It is usually single layered, but sometimes it is multi-layered in Nicotiana tabacum. Nicotiana tabacum, this endothelium layer is called multi-layered structure. You may recall that endothelium originate from the primary pericytial cell. This type of endothelium can mostly differentiated in the four protuberant of the building part of the anther. Sometimes it also develops near the connectivity region of the anther. In some plants, like tetricales meaning wheat like this, on entire ring of endothelium is present. This is the another special character of anther. The endothelium cell are rapidly elongated. Their full development is attained by the time of the anther is ready to dehiscence or to discharge of the pollen grain. At maturity time, the endothelial cells can be easily recognized by the process of fibrous bands that usually arise from the inner tangential wall. The fibrous bands radiate to outwards and upwards, ending near the outer wall of the each cell. Hence, the inner tangential wall appear in thick, whereas the outer tangential wall appear in the thin. These thickenings contain a high proportion of the cellulose and small amount of the pectins are present. As mentioned above, the endothelial cells, owing to their thickening, help in the dehiscence of the anther. This is brought about to the differentiated from the hygroscopic expansion of the outer and inner tangential wall of the endothelial cells. You may recall that the inner wall are thicker due to the presence of more alpha cellulose than the outer ones. Therefore, the inner walls absorbs more moisture and expand more than the outer ones. This result in the rupturing of the anther wall. That's where the rupture or opening up anther occurs. Now the anther very weak point of the anther. That is areas in the anther lack thickening or the longitudinally dehiscence of the anther, which are most common occurrence in there. There are usually two such weak areas. These are also some variations in the endothelium structure and function. In the clistogamous, this is a one type of flower development whose never open. Clistogams meaning the flowers never open. This is the type of development. This is the that type of flowering is called the clistogamous. And endothelial fibrous thickening are absent. These cells may also not develop in plants whose anther descends by apical pores as the dehiscence of anther takes place by the contained apex of the anther layer. This is called the endothecium. The second layer also completed. The third layer is called middle layer. The third one is called the middle. Inner to the endothecium lie one or three layers of the cells, which are collectively termed middle layer. These originate from the primary pericytial cells and have a nutritive function. The cells compressing these layers are generally rich in reserve food material, such as the main reserve food material is starch grains. During early stages of anther development, these cells are generally ephemeral. When the microspore mother cells undergoes meiosis division, the cells of middle layer usually become flattened and crushed, composed of the single layer cell. These cells are completely surrounded the sporogenous tissue, and they attain maximum development when the sporogenous tissue is at the stage is called the tetrad stage. The tapetum is dual origin and dual origin of this one. This dual origin uh, the attain, that is the dual origin tissues or the tapetum. Most of these cells uh, compactively, that one is the usually a difference in the structure. The tapetum dual role, the tapetum dual role is this one. Usually connective tapetal cell differ in the dimorphic tapetum, and the usually origin and appears different in size structure. The tapetum of dual origin is known as, that is called the dimorphism tapetum. In Alectria thamosium, this one is tapetum origin is cells are outer side are much smaller than inner side. The size also different. And the next coming to the tapetum is the nursing tissues. That is called the this one is the tapetal cell remain contact with the microspore mother cell. That is called the MMC. Microspore mother cell is called the name is called MMC. This MMC as a soon meiosis is completely completed these connections become a plugged. Connections become a plugged. And tapetum also play an important role and formation of exine and intine. This deposition tryptophan in the pollen kit. This is called the tryptophan. You will know more details about the two terms of the next unit. We shall now study the structure of the tapetal cells. To begin with, the tapetal cells are dense cytoplasm. Each and every cell dense cytoplasm and unique uninucleated structure. They often become they often become from two or four lobed structure. Even here 16 nucleates also present into the this one. And the multinucleate conditions. This is the multinucleate condition. Multinucleate conditions result in the form of nuclear divisions, not accompanied by to the cytoplasmic division. Only for the nuclear divisions are present in this one. And next one, coming to the tapetal cells also become tapetal cells also become polyploidal either due to endomitosis or polyidentity and formation of restitutions nuclei. Thus, there is an increase in DNA content, but there is no chance in the number of chromosomes in the nucleus. For the normal development of pollen grain, the development of the tapetum is very important. If the tapetum fails, the development functional abnormal viable pollen grains are not formed. The pollen sterility result. And this one, the result, in angiosperms, there are principal type of tapetum. The amoeboid and the secretory tissues. In the secretory tapetum, the constant cells maintain their individual and position. Whereas in the amoeboidal cells, the amoeboidal cell type, the cells charge their position and shape during the ontogeny. We shall now study the few more details about to the tapetum also. Two types of tapetum is there. Amoeboid tapetum and the second one is called the secretory tapetum. Amoeboid tapetum, here the tapetum structure is not clear. In this one is a shapeless tapetum. That is not different. Structure is not particular. Here it is also invasive or periplasmodial tapetum. This is called the periplasmodial. This type of tapetum is more prevalent. It is more prevalent and this one is the monocotyledon plants. This monocotyledon rum. Then the dicotyledon plant, Helianthus. In the amoeboid tapetum, there is a breakdown of the inner tangential cell wall followed by the enlargement and movement of protoplast into the anther sac, where they fuse and form periplasmodium. The formation of periplasmodium occurs at different development stages, ranging from meiotic purpose and to form to the tetroid stage of the pollen development. in different species. In anther they fused. In anther they fused into protoplast, periplasmodial, closely surrounded by the developing microscopes playing into the plan important role for the pollen development. At the tetroid stage, the tapetal cells cytoplasm produced the enzyme, that is enzyme is a callase, which breaks down to the callus. Callus wall around the microspore tetroids, resulting the microspore into the periplasmodium towards the end of the pollen development. The periplasmodium degenerate, dry up, the become coated surface in the pollen wall. This is the amoeboid tapetum. Towards the end of the pollen development, periplasmodium degenerate, dry up and become coated surface of the pollen wall as pollen kit material. This is the amoeboid tapetum. Amoeboid. This shape, this is called the amoeboid. Next one is the secretory tapetum. Secretory tapetum has other name partial cellular or granular tapetum. Secretory tapetum is more commonly observed in the dicotyledons. Unlike the amoeboid tapetum, the cells of the tapetum remain intact in their original position till the maturation of the pollen grain. The tapetal cells, these create the liberate substrates to the anther sac from their inner walls. In several taxa that are characterized by the creator tapetum, there is deposition of sporopollenin granules on the inner face of the tapetal cell. These granules referred to as the orbicular, that is the ubisco bodies. These can be easily seen in the light microscope. These orbiculars generally absent in plants like Plasmodial tapetum. The orbicula originate in their cytoplasm as lipoidal pro-orbiscular bodies, ubisco bodies with a limiting membrane. These pro-orbiscular bodies accumulate below the plasma lemma and are extruded to the cell surface where they acquire sporopollenin coating. It has been believed that the orbicula have a role in pollen exine formation and pollen dispersal. The second, another tissue is called sporogenous tissue. You may that primary PPC is the primary pericytial cell. Second one is the primary sporogenous cell. Primary sporogenous cells result after the pericytial division of the archisporial cells, the PSC may undergo mitotic division and increase in number before functioning as the MMC, and PSC may function as MMC is undergoes, that is a MMC meaning microspore mother cell. This microspore mother cell undergo meiosis division and gives rise to haploid microspore. The detail of the meiosis have been extensively studied. Anther ideal system studied in the meiosis. This is mainly due to the fact that they are radially available and accessible for experimentation, and each anther contain a large number of meiocytes mostly exhibit in synchrony. Most of information meiosis has been obtained by from investigation on the anther. Many increasing studies have been carried out. And the next, what is the many increasing studies have been cleared out the meiosis is anther by using tissues and organ culture technique. It is possible and remove anther at different stages of development culture. Then on a nutrient media investigation, microsporogenesis, when the anther are before excited before the initiation of meiosis and cultured on a medium. Meiosis is not initiated is not initiated that one of the division contain cultured anther. Result shows the anther oh, require a special uh, apart from basic nutrient for the initiation of meiosis. What is the exact the next one, meiosis. Meiosis is the significant event in sexual reproduction. You recall from study of unit 3. That is the apart from reducing E60. And that is the apart from reducing diploid chromosome number meiosis and the variable recombinant and containing crossing over. This is the event leads to genotypic combination. This is the insertion of the sexual reproduction. As you have studied the mitosis cycle, mitotic cycle can be divided into phases. Meiosis 1 consists of a reduction division resulting in two haploid cells, nuclei. Hmm. And the next one, this is the structure only wall. Next class we will discuss about to the complete structure of the uh, anther. Today class we will discuss medicinal plants. Okay.